Hello, and thank you for joining today's cutting-edge conversation about the challenges and innovations in biomarker research. We appreciate you joining and looking forward to having a lively discussion among our esteemed panel and with you, our audience. The session is being recorded and the materials will be provided after the webinar. We will begin today's webinar by introducing you to our speakers. I'll kick it off with a high-level introduction to biomarkers, and then you'll hear a presentation from each of our panelists about a particular approach or technology to support biomarker research. We'd also love to hear from you throughout this presentation. So if you have a question for any of the panelists or myself, please feel free to submit them using the Q&A function, which you should see at the bottom of your Zoom window with the large letters Q and A. Last but not least, if you're interested in getting in touch with any of the speakers after the webinar, their contact information will be available on the last slide, and you can access each organization through Scientistscom's marketplace. Now, I'll begin with the introductions. So my name is Matt McLaughlin, and I'll be your host today. I am the Senior Vice President of Compliance and Categories at Scientistcom. I've been involved in drug research and discovery for over 20 years, and for 13 of those, I was a research scientist at AstraZeneca in the R&D genetics and personalized healthcare biomarker departments. During my time there, I was responsible for a global Sanger sequencing and SNP genotyping core capability, which delivered a number of pharmacogenomic projects, such as determining the influence of cytochrome P450-TC19 genotype on platelet function in patients treated with ticagrelor versus clopidogrel. Now, for one of my roles within Scientistcom is to drive the global strategies regarding biomarker and clinical research to ensure the platform provides enhanced functionality and benefit to our users. But more importantly, let's introduce you to our speakers and panelists. First, we have Morad Nakli. Morad joined Owlstone in 2018 and performs as head commercial biomarker strategist. In his role, Morad supports the development of scientific and technical solutions based on exhaled volatile organic compounds analysis for clinical applications. Morad has over 11 years of experience in biomarker discovery and exhaled breath analysis. He holds a first degree in emergency medicine, a master's degree in medical sciences, and a PhD in nanobiotechnology. Prior to joining Owlstone, as part of his postdoctoral research, Morad focused on investigating the origins and biochemistry of VOCs, mainly in respiratory diseases. Next, it's Bharvika Patel from Lanterne DX. Bharvika received her BS in psychology with a minor in molecular and cellular biology and chemistry from the University of Illinois. She later went on to complete her PhD in neuroscience from Iowa State and is currently a scientist at Lanterne DX. Bharvika has over 7 years of experience with immunohistochemistry, fluorescent microscopy, and now support Lanterne DX multiplex imaging and digital image analysis using various platforms. And last but not least, Natalie Smith, the Senior Pharma and Translational Genomics Manager at Eurofins Genomics. Natalie has over 18 years of experience in managing technical projects with large international scientific companies, specializing in pharmacogenomics, bioinformatics, pharmaceutical drug release, and genomics technologies. Her background includes several technical certifications, including FDA pharmacology and toxicology licensing. She's worked for Eurofins Scientific in both the US and now for the European division called Eurofins Genomics, where she is based in the UK. Thank you to our panelists. Now let's crack on. I thought it'd be a good place to start with the basic definition of what a biomarker actually is, but this proved a bit more difficult than I first anticipated. Over the years, definitions were generally similar, but varied on the specifics. However, in 1998, the NIH Biomarkers Definitions Working Group defined a biomarker as a characteristic that is objectively measured and evaluated as an indicator of normal biological processes, pathogenic processes, or pharmacologic responses to a therapeutic intervention. This group brought together leaders in the field of clinical trials and biostatistics from the NIH, the FDA, academia, and industry to develop consistent and comprehensive definitions of terms relating to the use of biomarkers as surrogate outcomes. And these definitions are now frequently cited in the literature. For me, though, when discussing biomarkers, that can be a bit dry and doesn't put the impact into context. So for me, a biomarker in the drug development context can either be a molecular, cellular, or imaging based. It's used for either predictive, prognostic, or diagnostic purposes, and it's intended to move us away from a one-size-fits-all approach to ensure that we are treating the right patient with the right drug at the right dose, using the right route and at the right time. That got me to thinking, so how long biomarker research has actually been an area of interest? And when I was looking through the research, I realized that it's actually a lot longer than I first thought. I came across the work of Hippocrates some 2,500 years ago, describing the individuality of disease and the necessity of giving different drugs to different patients. And more recently, in the 1950s, the term biological marker first began to be used. However, it took another 30 years for biomarker itself to become common. And then from there, a further 20 years to actually reach the definition developed by the working group described on the previous slides. Outside of the literature, though, people refer to and use biomarkers every day without necessarily knowing. Body temperature can be a biomarker for infection, measuring blood pressure can be a biomarker for risk of stroke, and cholesterol can be a biomarker for increased risk of coronary disease. However, as medicine and precision medicine come to the fore in the past 20 years, biomarkers are now much more complex and challenging to be measured reliably and consistently. Of those, oncology has been at the forefront, and one of the most well-known examples is related to the human epidermal growth factor receptor 2 oncoprotein, more commonly known as HER2. Discovery of the HER2 oncogene led to the development of one of the first approved humanized monoclonal antibody-based targeted therapies, which I can never pronounce, but it's trastuzumab. But helpfully, they've also named it Herceptin, which revolutionized breast cancer management. So biomarkers are clearly important, and I believe are critical to the future of drug development. They enable new strategies in the development of better drugs for patients. So what is Scientist.com doing to support scientists in their research? Well, as we don't conduct the research ourselves, we have focused on supporting activities and developed several tools exclusively available via the Scientist.com platform to increase access to information and therefore speed up quality decision-making within the area. Via the marketplace, researchers and suppliers can now proactively access biomarker-specific technical RFIs to better understand and communicate capabilities. We've launched our new Comply RFI to assist clients in understanding a supplier's policies and procedures prior to engagement to ensure they work with the best quality suppliers. And we've also partnered with Equipped, which stands for Enhancing Quality in Preclinical Data, which came out of an IMI initiative to make available their quality standard regarding preclinical data. So rather than trawling Google for information or completing endless questionnaires on behalf of clients, the marketplace is now your one-stop shop for relevant biomarker information to assist research. But this access to information and simplifying the sourcing process aren't the only challenges in the space. Over the years, the same challenges impact researchers across the globe as they aim to develop new treatments for patients. There is the desire to discover new biomarkers, to measure existing biomarkers using new technologies, and to increase efficiency and efficacy of measuring biomarkers. As research progresses, these core challenges will remain, and it's up to researchers like you on the webinar and the companies like our panelists working together to overcome them for the better of humanity. So now the scene is set, I'm excited to hand over to the panelists for their presentation to provide you information on the work they're doing in this exciting area of research. Our first panelist will cover the breath biopsy platform from Owlstone Medical, and it's my pleasure to introduce you to our speaker, Morad Nakli.
Thank you very much, Matt, for the kind introduction and the opportunity to provide an overview of Owlstone Medical and our development of solutions and technologies in the space of exhale breath analysis. And that's all towards the development of non-invasive disease detection and precision medicine. Now, I was really intrigued by your introduction, and I wanted just to state that Hippocrates was also the first to be cited, telling his students to smell their patients and look for clues for diseases. And this is the first indication in literature that volatile compounds could be biomarkers for diseases. So that links really beautifully. In the next slide, yeah, please, thank you very much. So our mission at Oldstone Medical is to save 100,000 lives and 1.5 billion in healthcare costs. And that's through the commercialization of clinically valuable breath-based tests that would be integrated in the clinical practice for the early detection of diseases, as well as in precision medicine. Owlstone is established as the leader in the field of breath biopsy with over 15 years of experience in volatile compounds detection and research. We have a multidisciplinary team of almost 200 employees, including scientists, engineers, and software developers, with sites in Cambridge in the UK and North Carolina in the States. We have worked with over 100 different customers from both the academic sector and pharmaceutical industry, as well as participating in key initiatives, including the Horizon 2020 and 3TR. Now, in addition to providing research products and services to our customers, we are also developing a range of novel breath tests for the early diagnosis of lung cancer, for liver diseases such as NASH and cirrhosis, and other respiratory diseases in partnership with pharma companies. We have recently launched our first digestive health test based on hydrogen and methane breath testing service in partnership with the Functional Gut Clinic in London for the diagnosis of gut health problems. Our focus is the volatile phase of exhaled breath, namely volatile organic compounds, or VOCs, as I will call it in these slides. What many don't know is that exhaled breath contains hundreds of VOCs that are from indigenous source. Those are products and byproducts of biological and metabolic processes that after their circulation in the bloodstream and due to their volatility, they easily diffuse through the lungs to exhaled breath where they are detectable. In addition, we are continually being exposed to compounds of exogenous sources, such as medications, diet, inhaled pollutants, and others that also end up being exhaled either in their original form or their products following enzymatic metabolism. Therefore, exhaled breath is a very rich biological matrix containing compounds that reflect on intrinsic metabolism, allows estimation of exposure levels, and assessment of specific metabolic pathways in the body. And indeed, breath biomarkers actually exist, and they're already in clinical use. Carbon dioxide, carbon monoxide in Judith, hydrogen and methane for lactase deficiency and small intestinal bacterial overgrowth, nitric oxide in asthma, volatile sulfuric compounds in halitosis, and a group of exhaled alkanes that indicate heart transplant rejection and others. Today, we also know about hundreds of other volatile organic compounds with established association with metabolic pathways and processes, such as inflammation, oxidative stress and lipid peroxidation, glucose metabolism, liver enzymatic activity, bacterial activity, and others. Therefore, exhaled volatile organic compounds could fit in a wide range of clinical applications. They could be used as non-invasive diagnostics for early detection and screening, or as companion biomarkers in personalized medicine. Exhaled breath in general offers a safe and easy-to-use point-of-care and home testing applications. It allows monitoring of occupational exposure levels to hazardous substances, for example. And in general, it's a very valuable research tool to assess mechanism of action, dynamics and kinetics of drugs during development. Now, to do so, we develop and commercialize end-to-end solutions that allow reliable capture and analysis of VOCs. On the collection side, we have introduced the receiver breath sampler, the first standardized, reliable, and reproducible breath sampling device, in conjugation with Casper portable air filter, providing low background for sampling, minimizing environmental impact and reducing the variability. Controlled by the breath biopsy collect software, receiver accurately capture specific fractions of exhaled VOCs on designated cartridges. Those allow storage and shipment of samples for the analysis. We have also developed the Omni assay, our signature analytical platform, designed to provide the broadest and most reliable global on-breath VOCs analysis. For the Omni assay, we choose to use gas chromatography linked with high-resolution mass spectrometry, optimizing the sensitivity, the dynamic ranges, and the ability to identify novel biomarkers in breath. We also provide comprehensive support to our customer studies, ensuring streamlined integration into clinical and research trials, including customized study design, power calculation, protocols, support for site initiation, installation of the breath biopsy station, training to the staff. We provide site monitoring and clinical project management, and of course, at the end, the statistical analysis with meaningful clinical and biological interpretation to answer the key scientific and clinical questions. And therefore, every step in the workflow was tailored to optimize the signal to background ratio to minimize the variability of the data and the risk of false discovery. And that includes the sampling, the sample handling, to quality control measures, analytical stability over time, and even the data processing itself. And thus the workflow allows reliable investigation of hundreds of different volatile organic compounds in breath samples and provides a meaningful biological interpretation, maximizing the chances of success on those clinical trials. To assess the capabilities and the performance of the Omni assay, we routinely perform what we call the BOP study. That stands for breath or blank. In this particular case, we obtained 57 breath samples from 4 different volunteers over the course of 4 different days and obtained equal number of representative blanks. Those blanks are meant to quantify any background VOCs that might end up in breath sample. However, they might not be coming from breath itself. We use those to establish the signal to background of VOCs and make sure that each individual volatile organic compound is at least 3 standard deviations above the corresponding blank concentration. And that gives us the confidence to say it's actually coming from breath, and this is how I can link it to biological pathways. In this study, we detected over 1,450 VOCs found in different breath samples, and we found a median of just over 500 on-breath VOCs per sample. In terms of repeatability, we saw over 260 on-breath VOCs that were detected across at least 70% of the samples, and over 400 on-breath VOCs that were detected across at least 50% of the samples. The intra-subject variability was measured by the median relative standard deviation of on-breath VOCs, and it ranged between 26 to 36% in the different volunteers. And the overall study variability, that includes the biological variability, the pre-analytical and analytical variability, was 49% when assessed for over 390 volatile organic compounds. Now, to monitor and correct for analytical variability, each sample is injected with 8 internal standards. And the variability of those internal standards recorded over the period of month for this analysis is listed here in the table, and you can see it ranging between 3% to 13%. We also run over 50 calibration curves per day on the platform of compounds typically found in breath, and that allows us to monitor the performance and suitability of the platform in terms of sensitivity, limits of detections, linearity, and so on. Now, as I mentioned previously, our ultimate goal is to commercialize clinically valuable breath-based tests, and here I present our end-to-end test development pipeline. So following the discovery of candidate biomarkers, we developed bespoke hardware and targeted analytical assays to allow clinical validation of the biomarkers in the intended for use population as a key step towards the commercialization of any clinical test. And one example here is the hydrogen and methane breath test for small intestinal bacterial overgrowth we recently launched with the Functional Gut Clinic. Thank you very much for listening. And I will be more than happy to answer any questions during the discussions or follow up through email.
Thank you very much, Morad. I found that fascinating and love how non-invasive the solution that you guys have developed is. In the interest of time, moving on, as Murad said, please don't forget to submit your questions via the Q&A functionality. But our next speaker is Bhavika Patel from Lanterne. So over to you.
Thank you for the introduction, Matt. As Matt said, my name is Bhavika Patel, and I'm a scientist at Lanterne DX. Today, I want to share the benefits of multiplex immunofluorescence and how it can be a valuable tool for researchers and clinicians. In today's talk, I will speak about how we can use multiplex immunofluorescence to better understand the tumor microenvironment in an effort to develop new strategies for immuno-oncology. But first, many of you may not be familiar with Lanterne DX, and I would like to give a brief introduction into who we are and what we do. We are a clinical histopathology CRO based out of Boulder, Colorado. Our team of expert histotechnicians, pathologists, and scientists bring over 20 years of experience in histology, pathology services, and digital image analysis in pharma, biotech, and academic industries. As a CLIA-compliant facility, we offer numerous services such as histology, biomarker discovery, pathology support, and digital image analysis. Lanterne DX offers histology support services such as processing and embedding, tissue sectioning, and special stains. We also offer biomarker discovery services in which we optimize antibodies for IHC applications. We additionally provide KISH and FISH for RNA analysis and can develop multiplex panels, which includes multiple IHCs, KISH, or a combination of both. All of our biomarker discovery efforts are guided with the MD board certified pathologist who can perform manual analysis for diagnostic histopathology, as well as offer support for digital image analysis. But today I will be speaking about why biomarkers are important and how multiplexing can help advance a client's understanding of the tumor microenvironment. I will also briefly touch upon some ready-to-use algorithms we have available that provide various outputs such as co-localization results and phenotyping results. So why are biomarkers important in both the research and clinical settings? Well, biomarkers are a widely used tool for both research and diagnostic applications. It can expand our understanding of how drugs work, the efficacy of drugs, as well as guide clinical decisions. In our workflow, we use biomarkers at multiple different stages, including initial method development, and for validation and assay development. Ultimately, our goal is to move our clients into clinical stages where we can help further validate their biomarkers for diagnostic, prognostic, and treatment strategies. Oftentimes, there may be multiple biomarkers that need to be initially evaluated for use, and this is where multiplexing is valuable. The ability to label multiple markers within a single tissue can provide valuable information about the complex tissue microenvironment. The tissue microenvironment is characterized by complex immune phenotypes and tumor evasion mechanisms, which are constantly changing. Compared to conventional immunohistochemistry, which is a widely used diagnostic technique in tissue pathology, multiplex immunohistochemistry allows us to label multiple markers in a single tissue, enabling researchers to quickly interpret the complex phenotypes of the tumor microenvironment. And this helps advance research discoveries. Multiplexing immunofluorescence also helps preserve spatial context, which allows us to better phenotype cells within the tissue. With our collaboration with Akoya Biosciences, we have utilized their Motif PD1, PDL1 panel kit to stain lung tissue. Our workflow involves staining using the Leica Bond RX autostainer system, followed by whole slide multispectral imaging using the Vectra Polaris. As you can see, we perform whole slide imaging, allowing us to capture data in both the tumor and the tumor microenvironment fields. Once we have these images, we can input them into the Inform software to follow a predefined phenotyping algorithm, which allows for phenotyping and spatial analyses of our tissue. The PD1, PDL1 lung cancer panel kit is designed to detect 6 clinically relevant biomarkers in lung cancer FFPE tissues. These biomarkers are FOXP3 for regulatory T cells, the PD1 ligand PDL1, PAN-CK for epithelial cells, the T cell regulator PD-1, CD8 for cytotoxic T cells, and CD68 for macrophages. This is followed by a nuclei counterstain. Using these 6 biomarkers, we stained and imaged non-small cell lung cancer tissue. And as you can see, the PD-1, PD-L1 panel kit allows for 7-color multispectral imaging of whole slides. In this panel, FOXP3 is yellow, PD-L1 is green, PanCK is red, PD-1 is orange, CD8 is cyan, and CD68 is magenta. From these specific regions of interest within the non-small cell lung cancer tissue, we can use the predefined algorithm in the Inform software to analyze phenotypes of cells. Here, we are using the same algorithm across multiple different lung cancers to quantitate the number of cells expressing each of these biomarkers. In A, we show data for non-small cell lung cancer tissue. In B, we show lung adenocarcinoma. And in C, we show large cell lung cancer. Our data shows that different populations of cells exist in all 3 of these tissue indications, allowing researchers and pathologists to quickly and efficiently decide optimal protocols for diagnostic purposes. Furthermore, this data can help us interpret what cells make up the tumor microenvironment in each of these lung cancer cases. After phenotyping the cells, we can perform more advanced analysis, including spatial analyses using Phenopter reports. This helps us better understand cell-to-cell interactions within the tissue. These figures are called touching cell plots, in which a cell is outlined and filled in if it is touching a cell of a paired phenotype. In this example, we use 2 phenotypes, PanCK and CD8, to determine where T cells are relative to tumor. The PanCK marker detects tumor epithelial cells, and therefore this algorithm can provide insight into how T cells interact with the tumor microenvironment and provide insight into tumor infiltration. Furthermore, we showed that the algorithm can be utilized for other lung cancer tissues, including lung adenocarcinoma and large cell lung cancer. By utilizing the same algorithm, we're able to analyze multiple lung cancer tissue types, showing the adaptability of the Motif PD-1, PD-L1 lung cancer panel kit. Our final question was if the Motif PD-1, PD-L1 lung cancer panel kit can be used for tissues other than lung tissue. The simple answer is yes. Although the kit was designed for lung cancer tissue, Lanterne DX was able to utilize the kit to analyze tumor microenvironments within different types of cancers, making the Akoya Motif PD-1, PD-L1 panel kit a powerful tool. Here, we see the kit can be adapted to stain a tissue section obtained from head and neck squamous cell carcinoma, breast cancer, and colon cancer. We were able to use the same 6 biomarkers to look at the immune phenotypes across different tissue types and are continuing to test this panel in additional tissue indications. In conclusion, we show that the non-small cell lung cancer, lung adenocarcinoma, and large cell lung cancer, we can identify populations of CD8, PD-1, FOXP3, CD68, and PanCK positive cells, as well as PD-L1 localization. This analysis also helps us better understand cell-to-cell interactions using the predefined spatial analyses algorithms. CD8 and PanCK cell-to-cell interactions help us understand how T cells interact within the tumor microenvironment and give us insight into tumor infiltration. The Motif PD-1, PD-L1 panel provides unparalleled quantitative data for preclinical and translational immuno-oncology research. In summary, Lanterne DX utilized this ready-to-use Motif PD-1, PD-L1 lung cancer kit to expand across multiple tissue types, including head and neck cancer, breast cancer, and colon cancer. We are currently expanding tissue indications as well as sample size to produce reproducible data using a single algorithm. Our current goal is to utilize the VisioPharm platform to conduct image analysis using deep learning algorithms to identify tumor regions. Overall, multiplexing immunohistochemistry is a powerful tool for the development of strategies for immunotherapy and precision medicine. Thank you all for listening, and I'll be happy to take questions during the Q&A.
Thank you very much for the presentation and the excellent work. I remember back in my day when I used to wear a lab coat, the multiplexing of IHC was very early in its day, so it's nice to see how it's progressed. Our next and final speaker is Natalie Smith from Eurofins Genomics. Remember, everybody on the webinar today, you can submit your questions for all three presenters via the Q&A box, and they'll answer them during the last portion of the webinar. But for now, over to you now, Natalie.
Thanks. Hello, and welcome to Eurofins Genomics presentation titled The Integrated Genomics Universe from Biomarker Discovery to Biomarkers within Clinical Trials. Next slide, please. My name is Nat. That's going to be the previous slide. I'm sorry, I've got something in my throat here. Thank you. So my name is Natalie, and today I'll be talking to you about the capabilities of Eurofins Genomics as they relate to biomarker discovery and clinical trials experience. I will also touch on our general portfolio capabilities. Next slide, please. Eurofins Scientific is an international life sciences company. We offer a vast network of complementary scientific labs located around the world, allowing you to have all of your project work done under one company, with fewer contracts and points of contact. We have over 200,000 validated methods in over 50 countries with 900 laboratories. Next slide, please. Eurofins Genomics offers a full suite of genomic services that cover work including DNA and RNA extraction, gene synthesis, custom sequencing, applied genomic services like oligos and Sanger work, and a full suite of arrays technology platforms. Once the project is complete, we also have expert bioinformaticians that can help you to make the most of your data. Next slide, please. As part of our NGS services, we can offer you immediate access to innovative NGS solutions, professional development, implementation and validation, immediate reliable support with technical expertise, dedicated project management and consulting for customized solutions. We perform all of our work to ISO 17025 standards, and in addition to this, we offer the full scope of compliance levels for ISO 13485, GCP, GLP, and GMP for selected specific services. For microarray service, we provide accredited standards to ISO 17025. We are also GLP and GCP compliant. We have high throughput and custom arrays options, the latest technology platforms, and dedicated project management and consulting for customized solutions. Next slide, please. With our NGS and Array Solutions, we can support your every step of the way through your project. For genomics projects, we offer whole genome sequencing to detect single nucleotide variants, insertions, deletions, copy number changes, and large structural variants. We can offer greater than 30x coverage with 150 paired end sequencing on the NovaSeq 6000. We can also provide low-pass whole genome sequencing with greater than 10x coverage, which is also run on the NovaSeq 6000. We offer genome-wide screening on the Illumina Global Screening Array and the Precision Medicine Research Array and Precision Medicine Diversity Array from Thermo Fisher. For epigenetics, we offer whole genome bisulfite sequencing, which is regarded as the gold standard technology for detection of DNA methylation because it provides qualitative, quantitative, and efficient approach to identify 5-methylcytosine at single base pair resolution. We also offer low-pass whole genome bisulfite sequencing, which is cost-efficient and a good alternative to whole genome bisulfite sequencing. For microarrays, we offer the Infinium Methylation Epic Array that allows the integration of methylation patterns at the genome-wide level. Covering more than 850,000 methylation sites, this array is very popular and is an ideal solution for epigenome-wide association studies, even for those tricky FFPE samples. Next slide, please. So here we take a look at the focused genome-wide biomarker discovery. It consists of two major steps, transcription and translation. As we know, together, transcription and translation are known as gene expression. For the DNA level work, we provide exome sequencing, high coverage cancer panels, customized panels, and microarrays. For transcription level work, we offer whole transcriptome analysis, RNA enrichment, and gene expression microarrays. For epigenetics projects, we suggest reduced representation by sulfite sequencing and target enrichment panels. Reduced representation sequencing, or RRS, is an approach to generate genome-wide high-throughput sequencing data and obtain a large number of genetic polymorphism tag sequences. This helps to fully represent the whole genome information of the species. We also offer target enrichment for regional focus. The genes of interest are enriched via proprietary Eurofins protocols using the latest adjuvant hybridization technology. The target enrichment system captures genomic targets using long RNA baits. The specialized protocol enables the generation of highly complex libraries, deep coverage of regions of interest, and improved sequence uniformity. Following target enrichment, next generation sequencing of the library is then performed. We also offer here the EPIC microarray, which I talked a little bit about earlier. Next slide, please. Here we show the next stages. So once you have found your biomarker signature, what are the next steps? So the biomarker validation process includes companion diagnostics, biomarker validation, drug development population screening, and also the development of diagnostic services. So you can use all of these full suite of services that we offer to complement and help you with every step. So some of the combining technique examples here would be NGS, qPCR, digital PCR, Sanger sequencing, and microarrays. Next slide, please. So depending on your project, we can offer a wide range of services to cover your areas of interest for biomarker projects. Here we have highlighted Sanger, qPCR, midplex, microarrays, NGS, and exome sequencing. To complement your project, we can also perform the isolation of new material and the bioanalysis of your data afterwards. Next slide, please. With our NGS and Array Solutions, we can support every step in your entire value chain. Here we show examples of the services we can offer to help you with your biomarker projects. These services can cover NGS, arrays, and applied genomic technologies to produce meaningful data for whole genome sequencing, custom panels, and specific gene SNPs. We can also help you to isolate the material, like I talked about before, and the bioanalysis to help you either in the setup process or to analyze the data afterwards. Next slide, please. So Eurofins can help to provide intercompany disciplines that span the full spectrum of R&D and interclinical trials for genomics-related technologies. We provide dedicated project management with every service and expert advice as required. We can offer many high-quality service levels, including GCP, GLP, GMP, and ISO 17025. We have a wide range of expertise in early phase clinical trials work and can work in accordance to the FDA guidelines. With a portfolio covering NGS arrays, Sanger, and PCR services, we can find a solution to help you in your progression from discovery through the clinical trials phases. Next slide, please. So thank you everyone for your attention today, and I look forward to answering your questions afterwards. Thank you, Matt.
Thank you very much, Natalie. So that concludes our presentations for this portion of the event. So what we're going to do now is move into the Q&A section. So if you do have questions for the panelists, please, if you go into your Zoom window, you'll see a Q&A section or Q&A icon at the bottom. You can submit your questions through there. One thing that I would ask, if you do want to ask a question that's specific to a particular company or panelist, please don't forget to tag them. Otherwise, I may ask that question to somebody who you didn't want the answer from. But it could still be an interesting answer. It will just make sure you get the response you're after. So looking at those, the first question that I can see is going to be heading towards Owlstone. So Mohad, the question is, the breath biomarkers that you described and talked about in your presentation, are they applicable to preclinical species?
Yes, and it's a great question. So yes, we have proved, and the literature also shows that cell culture, for example, which we work with, when we sample the headspace, which is the gas trap above the media, it contains volatile organic compounds. We did actually a really nice proof of concept to correlate a class of volatile compounds, aldehydes, with inflammation and lipid peroxidation. And by simulating with hydrogen peroxide, for example, you could see the production of aldehydes above those cells. We also developed a lung and liver on chip models to allow tests for specific cell lines in an environment that is much closer to the lungs, for example, compared to a cell culture. And we have recently developed capabilities for animal model testing with mice, for example.
Thank you very much, Mohad. Hopefully that answers the question. Moving on, we have one now for Lanterne. So what improvements have you seen in the deep learning algorithms made available that you've used based on your presentation?
Yes, great question. So currently we use Visiopharm as our platform for image analysis, and they actually have a lot of ready-to-use algorithms, which are really handy because they have undergone a lot of training, and so it makes just you manually having to change the algorithm a little bit easier. And I will say the more training you can devote to an algorithm, the better the algorithm is. So it is a little bit of labor intensive in the beginning of developing a new algorithm, but it's worth it in the end. And I know Visiopharm also uses neural networks, and these are very adaptable and can be changed. And we can change the rules of the algorithm, which conventional image analysis does not really allow. And so these, the deep learning algorithms can definitely improve the accuracy in separating like tissue compartments.
Thank you very much. Questions are still coming in, so thank you for the submissions. Moving on to Natalie, one for you, and this is something that has brought back some many horrendous memories for me. What's your recommendation for strongly degraded FFPE RNA?
So with FFPE, it's a little bit harder when it's degraded. What you put in is generally what you get out. However, we do process FFPE samples all the time. And what we recommend here is to use the transcriptome service with ribodepletion. So then you can get better data back from that, even from that degraded RNA. Thank
you. And one thing just to add from a Scientist.com perspective, obviously, as Natalie says, what you get in, what you get out. So if you're looking to improve the quality of what goes in, then from a Scientist.com perspective, if you're ever struggling for samples, we have about 300 plus human biological sample providers spread across the globe that are available through the marketplace. They certainly have access to banks of FFPE samples, along with associated information on informed consent, ethical approvals and things like that. So if that's something of interest to support your research, feel free to reach out, and I'm happy to provide that information. But back to the topics of the presentation. So this one I'm going to actually throw over to Mohad again. When a patient, it's actually a dual question, so bear with me. First of all, how long do samples last in tubes? And then following on from that, when a patient breathes into the sample capture container, how long does the operator have to run it in the GCMS before it expires?
Thank you, Matt, for the question. Let's start with the first part. There is no real expiry date for the sample once you put it on the tube, because the risk over time for those tubes is either, if we think about it, either contamination or loss of volatile compounds from the tube. So what it might cause is mainly variability in the data. However, the protocols that we have developed, we developed to make sure that it doesn't happen or minimize that risk as much as possible. So in the first step after sampling breath, what we capture on the tube, in addition to the volatile compounds, is humidity, of course, in the exhaled breath. So the first step is to remove the excess of water, because the presence of the water, the oxygen, could impact the stability of the compounds on the tube. So first step, we flush it. We do what we call a dry purge process. We flush it with inert gas to remove the water and the oxygen remaining. Then we have a very specific hardware, the designated caps for long storage of those tubes, and we put them in at 4 degrees in the fridge, and that allows stability of the volatile compounds upon the adsorbent material for a few months. There are very nice papers there, publications that also tested the stability of compounds on similar tubes for up to 11 months and showed very, very nice recovery of those compounds with very small added variability, if any. Hope that answers the question.
Thank you, Mohad. And then to keep things moving along with everybody, I'm going to throw this one at Lanterne to give you a heads up. Can multiplexing be used as a diagnostic tool within your capabilities?
So that's an interesting question that we actually do get often. And the simple answer to that one is no, not yet. Although these kits are validated, they aren't validated to be used for diagnostic purposes. Most of our clients use multiplex immunofluorescence in retrospective studies to better understand how these markers can change in cancer tissues. They're also used as sort of, preclinical development phases if our clients are not really sure on which biomarkers would be of interest in their tissue. And so this multiplexing can really help with the preclinical phases of for some of our clients that are leaning more towards heading into clinical trials. But Akoya Biosciences is definitely working on getting these panels to be, used for diagnostics, because that's, the ultimate goal for both pathologists and clinicians and researchers.
Cool, thank you, Bhavika. Bhavika, sorry. Another question's just come in again for Owlstone. Is amplification needed for any of the volatile organic compounds that are less abundant?
So volatile organic compounds in exhaled breath are found in very, very low concentrations. So we do not perform any specific amplification, but the way we address those to make sure they are above the limit of detection when we run them on the instrument is by optimizing the volume of exhaled breath collected on the tubes. So this is the way we actually amplify it. And during the development of the Omni assay, we collected an increasing volumes on the tubes to capture finding that sweet spot between seeing those low abundant compounds on breath while maintaining a high quality chromatographic data, so we're not overloading the instrument itself. And we ended up with optimized sampling volume of 1.25 liters per tube, which takes on average about 13 minutes of a normal breathing into a receiver. And that actually figured that problem out. And now we see the low abundant compounds very clearly on the instrument.
Cool, thank you, Mohad. And Natalie, one for you now. What happens if their sample is not passing your QC? What steps would you take?
So with that, we would have a call with the client. So we would, if it didn't pass, then we would have a call with them. Usually we have our own standards to pass QC, and then sometimes we have client standards also that have to be met. So in most cases, it's not completely lost. We can rerun it. And so we generally call the client, let them know that it hasn't passed, but all is not lost. We can generally rerun those samples, and most of them are successful. So it just depends on if you have more sample left, if there's more that can be shipped, or if we can just rerun it, and sometimes that will save it.
Fantastic, thank you, Natalie. That's always good to hear. And over to the Lanterne team again. Can you talk a bit more about the process of developing a new multiplex panel? I know personally this would be something that would be quite interesting for myself, because as I said earlier on, multiplexing when I was back in the lab was still a pipe dream. But yeah, if you could share some more information on that.
Yes, of course. So at Lanterne DX, all of our multiplex panel development starts off with initially conducting monoplex immunohistochemistry, and that would be on our client's tissue of interest. And so once we do that, we do work with our pathologist to determine sort of the optimal starting conditions, at least for a monoplex IHC. And so once those conditions have been met and approved by the pathologist, we can then conduct monoplex immunofluorescence to better understand how the staining is working and making sure that using the same conditions for a chromogenic IHC match fluorescence IHC. And then from there, we use the data from both the monoplex IHC and the monoplex IF to then figure out which order we should place the antibodies in. And that's usually just determining which antibody has the strongest specificity and then the lowest specificity. And depending on that, the antibody is placed accordingly in a panel. So if the antibody has a lower specificity and lower signal, it's usually paired with a really bright opal if we're using the Akoya kit. And it usually would go first in the panel, so it's conducted first. And then if an antibody is probably the most consistent, we usually put that antibody towards the end of the panel. So we can definitely develop new panels. They are worth it actually in the end, because then you're able to actually utilize the panel for visualizing multiple different markers.
Brilliant, thank you. I found that fascinating. So we got one more question that's being asked. This is going to go to Owlstone. So, is there a need for background data on every patient for filtering the background in breath biopsy?
The answer is no. We don't need data for every patient because our internal testing and working with customers, we showed that the variability of the background, of course, it's there. But it's not too crazy that we need one per one. We currently recommend to our customers and even for our internal work, a ratio of 1:3, 1:5 between blanks and breath to capture and see the real distribution of the background and to allow us to determine what is of those signals we see on breath is real breath biomarker or not.
And actually, we just had another question come in. I'm just reading this to work out how to ask it. So I'm gonna open this up to the panel to see which ones you if any of you want to take this one. Would any of this platform work as a hybrid for diagnostic and personalized treatment protocol? Yeah, does anybody want to take that one? Go on, sorry, Mohad.
I'm happy to chip in and just stress what I mentioned in my talk previously. So we're currently assessing those biomarkers. I mean, it's at the end, it's a biomarker more than the platform because we could develop specific applications tailor-made for diagnostics, point of care, monitoring. It could be centralized and could be in clinic. That depends on the specific application and the positioning of the clinical test itself. So once we find those candidate biomarkers, we understand their biological rationale. We do the in vitro work and we develop the specific solutions in terms of hardware and the analytical aspect to meet the specifications of that specific clinical test. So yes, it could be a hybrid or it could be very tailor-made for a specific application.
Thank you. So with that, I'm going to begin the wrap up. So thank you very much for joining us for today's presentation. If anybody has any additional questions or would like to get in touch with our team at Scientist.com or any of the presenters, you can see our information available on this slide. The recording of this presentation will be sent to you by email within the next day or so. And I'd like to take this opportunity to thank our speakers for their involvement in the presentation and their dedication to the prep. I'd like to thank all of you for attending and joining us, and we hope that you found the presentation useful. This concludes the Cutting Edge Conversation webinar series, but we will be bringing it back next year, so please keep an eye out for that. And I'm likely not to speak to you again before the holidays, so I wish you all a happy rest of December and a joyful and hopefully less eventful 2022 when it arrives. Thank you very much and goodbye.